So our next speaker is Akshay Balsubramani from Stanford University, from Anshul Kundaji's lab, and he'll be describing their analysis of the DREAM challenge results on in vivo transcription factor binding predictions across cell types and from different methods.
So thanks, Arab. Can everyone hear me? All right. Great. So I'd like to first thank the RegSys organizers for giving me the chance to present this benchmarking challenge that we ran for predicting TF binding. And it was a collaboration with the ENCODE consortium and the DREAM challenges as well. Now, I mean, as everyone here knows, TFs regulates gene expression by binding to DNA, and there's a lot of experimental methods for assaying this in vitro and in vivo. But binding landscapes can vary a lot across cell types in vivo, and it gets untenable to assay sort of all the relevant TFs in the many cell types that are of interest. And so there's been many prediction methods for in vitro and in vivo TF binding. But although these have been evaluated systematically in vitro by a previous collaborative DREAM effort a few years ago, there's been no systematic sort of evaluation of these methods that's focused on in vivo binding. And so that's what I'm here to present. So let me just briefly go over the challenge design. ChIP-seq's the most popular assay for in vivo TF binding, so we use that to define sort of the ground truth here, irrespective of its caveats, which we can get to. We want to predict it basically from a minimal subset of putative determinants of binding and measured by some relatively commonly available assays and information. So this translates to, what we did is tile the genome with these 200 base pair windows, overlapping, label each window by whether or not it falls in a peak, and try to predict it from DNA sequence, DNA accessibility, as well as gene expression, and code annotations, and sequence-derived shape features, as was talked about yesterday. So what does prediction mean in our context? The most commonly sort of used formulation in the literature is this within cell type evaluation, where the training and test cell types are the same. And what changes is that you're just evaluating on different regions, held out regions of the genome. But the practical use of the desired ideal in vivo predictor is usually to scan genome-wide for binding sites in a cell type which has not been assayed yet. So we evaluate exactly that way, on a chromosome-wide scale and on held-out cell types, so cross-cell type prediction. And it's very different, again, from the common practice of subsampling the whole chromosome or whole genome for background regions and looking over those. So as far as the data goes, ENCODE's produced a huge quantity of data from which we carefully chose data sets with high quality, validated antibodies, reproducible peaks, some experiments that had never been released prior, which ended up being blind to the challenge participants and therefore were used for evaluation. And we ended up with a variety of cell lines as here, including a primary cell line, which was used heavily for evaluation, as well as blind held-out sets, as I mentioned before. And now to get into some of what happened in the challenge, we were really happy to have a large participant list, 40 teams submitted, and some of the very broad salient features of the top 4 algorithms are here. And I'll just sort of focus on 2 of them for now, which is the methods varied starkly in complexity, even though the performance ended up being quite similar for these top methods. On one hand, we have the method of Dan Kwong DQ here, a complex deep neural network which pulls features from raw base pair level sequence. And on the other hand, we have the co-winner YG, Yuanfang Guan, using sequence features derived essentially just from the canonical motif, not even trying to infer cofactors, but looking at weak hits and other derived features, and a pretty simple prediction model algorithm in the end. And there were some high-level design considerations that I obviously couldn't fit in that table over there. They're in the paper, but I'll get to them later if there's time. Now, as far as evaluating the challenge goes, the commonly used method of evaluation is, a measure of evaluation, I'm sorry, is AUROC. In this case, for this task, it's highly misleading. You can see it essentially seems to state that all the methods are doing well. Even more seriously, it fails to discriminate between top methods. Their rank orders are highly different. And the reason for this is, of course, because this binary classification task has very, very few bound sites, and the ROC metric is not very good for that. All the tasks in the challenge ended up being below 1% bound with our tiling of the genome. And so, precision recall performance is more informative. We used a few of those measures as well to aggregate the final ranking of methods in the challenge. And AUPRC is sort of a good stand-in for performance, as it turns out, along this entire curve. And we use that going forward. Another takeaway from this is that predicting across cell types is much more difficult than just within a cell type. Even though within cell type performance evaluation much more commonly used in publications, this is maybe not surprising. Regardless of the method, we did benchmark the top methods on within cell type evaluation as well, training as well, sorry, and the results are pretty clear. It's much easier. And another sort of aggregate view we can take is trying to see the effect of background. We can subsample the whole genome into commonly used background sets. And none of these directly represent chromosome-wide prediction, but evaluating any model on any sort of particular background subset of the genome introduces bias. So there's kind of two things to say here. The genome is really heterogeneous, so it is useful to split performance by background. A few of the common ones are shown here. Hope you can see them. Motif hits, DHSs, GC and dinucleotide matched regions as well as flanks. But it's useful not to bias, with any particular single background for evaluation. And the other thing you can see from here is that just looking at the raw AUPRCs, even on the top row, the methods substantially outperform the state of the art, but they still sort of underperform for most practical purposes, as we were talking about earlier, if you're looking from a downstream in vivo validation perspective. And so we want to get into the reasons for that performance, and that's basically going to be the rest of the talk. Now, the first thing we can do is ignore predictors for a minute and look at the data alone. These TFs are binding sequence, so we can plot motif enrichments inside the bound sites for each TF, each cell type that it's assayed on. Broadly, we see two types of factors. One is that there's occasional TFs like CTCF. Canonical motif dominates the landscape, and these are typically pretty constitutively bound across cell types. But by far, a more common situation is kind of like REST here, where you do have canonical motif dominating. It is a very long motif, highly specific binding, but we do get these other factors in here, even in this genome-wide enrichment signal. And you get even stronger variations on this for things like GIND and MAX. And just by observing this distribution of motifs across cell types, it's clear to us that there's a problem concerning cross-cell type prediction. Because if we naively train models on just training cell types and the sequence and fit them well through sequence, we'll tend to learn the features that are very highly characteristic of them, but they won't necessarily generalize across cell types. And this is the sort of main obstacle surmounted by the top performing methods in the challenge. And so we next want to investigate how these predictors behave in the presence of this varying cell type specificity of binding landscapes. So if we look at the bound sites, the binding landscape of a particular task, we can kind of generally divide it into these two extremes: constitutively bound in almost all cell types, and cell types specifically bound. And we can break the bound sites down by what other cell types they're bound in. But if we try to visualize this, then the naive way is a Venn diagram, just to see which subsets of sites are bound in different loci. But when several cell types are involved, it kind of risks looking like that, and many of you might have seen that before. Came in Nature a few years ago, but it can be pretty messy. And so rather than that, we'll just visualize these multi-way overlaps with a multi-way index like this, and this is breaking up the true bound sites for a particular task. Here we'll take CTCF in IPSCs. This was the easiest task in the challenge for all methods. And we can basically partition bound sites into these columns of this matrix, which are multi-indexes. And the top row is IPSC. It's all bound because those are the sites we're partitioning. And the other rows are different subsets of other cell types which have different binding statuses. This is in the style of upset R, if any of you are familiar with that package. But for this part of the talk, I'm kind of going to focus on the best predictor for each task and try to break down its predictions with respect to cell type specificity. So we can, for example, just plot in that first column for all sites that are constitutively bound across cell types, and look at how the best method, in this case DQ, the neural net that relies heavily on sequence, as we'll see. And it's clear that, number one, you've got 40% of the total bound sites being constitutively bound across these 10 cell types. And also, they are nearly predicted perfectly. Not too much of a surprise for CTCF to see that level of constitutive binding. There is some cell type specific binding. It is not predicted well at all. And most of the false negatives end up being cell type specific. So we can also, now that we've gone this far, plot other properties of these subsets, and DNase is one. Again, we can see here, just looking at the circled bits, that almost all the false negatives are essentially DNase inaccessible. They also happen to be in the cell type specific sites. And probably not a surprise, we can also plot each plot the motifs which discriminate each subset from the rest of the genome. And in this case, each subset is characterized primarily by the CTCF motif. But things get a bit more interesting if we switch tasks and look at, say, REST in liver. Another one of the final round tasks, best predicting method is YG. Does not, I mean, very simplistic featureization. Canonical motif relies heavily on DNase, and we'll see that in a second. Within cell type, most of the sites are cell type specific. They are not predicted well. Constitutive sites are predicted much better by the cross cell type version of this method. Within cell type training makes this cell type specific prediction much easier in a very discernible way. This is just the data being plotted. And we also see that when we plot the DNase, again, we see most false negatives, DNase inaccessible, but if you look within the constitutive sites, they are largely DNase poor anyway, but they are still predicted correctly because they're constitutive. The adaptation doesn't need to be through DNase there. We can go further and plot other things for that constitutive subset. Looking at this, we just break it down over chromatin states. We see that the constitutive one is heavily enriched for quiescent and polycomb repressed chromatin. We've got, that's sort of the canonical REST function. It's a prototypical repressor. And you've got also canonical motif enrichment when it happens in that subset, not in cell type specific sites, which don't really have very much polycomb repression happening. And we can go further and plot the motifs as we did before. Much more interesting picture here. Constitutively bound sites are the only ones which really are distinguished by the canonical motif from the background genome, as well as other cofactors. Canonical motif does not serve at all to distinguish other subsets from the genome. Instead, the two leading columns, two first columns are hepatocytic cell types, and we see HNF4 and FOXA, pretty canonical hepatocytic cofactors, characterizing those pretty well, and also further ones as you go further down the list. And we can switch cell types, not too much changes in terms of what we see. GATA distinguishes K562 specific binding sites. Again, in our, the rest motif is fairly, is distinguishing only of constitutive sites, and which are predicted nearly perfectly, again. So we look at this, and these top methods have very different ways of utilizing sequence and trying to surmount this problem, and we can try to see how they differ by turning this plot around a little, and instead of indexing by the cell types in which sites are bound, index by the the top methods predictions on sites, and break down all the sites which any top method predicts as bound. So if we break that down for CTCF, most of the predominant sites are where all methods predict bound. You get nearly perfect prediction. You can break them down by how many cell types are bound within them. It's constitutive for the most part, and highly DNA accessible. Now, we'd like to see how methods that differ in their ability to model sequence fare in cross-cell type prediction. And so we can, if we just expand out the plot, we kind of highlight this column here because we see that DQ, the best method on this task, as you can see, plots unilaterally, picks out some stuff that no other method can, with about 30% of them being bound sites. They're constitutive, and they're picked out in spite of being cell type inaccessible and very difficult for other methods, which is great. And when we switch tasks, we sort of see the other side of the picture. Jundi on liver here, very cell type specific binding landscape. YG does by far the best on this. It relies heavily on DNA's pretty simplistic model, as I said. And I've highlighted all the columns where YG predicts bound at all. They are the columns which are most enriched for bound sites, just looking at the top bars. They happen to have a lot of cell type specific binding sites, looking at that row. And they are also the ones which have many more accessible sites. Essentially, most of the other subsets are inaccessible. You can continue this and see these patterns elsewhere as well. And that pattern does very much, these patterns are very robust across all these tasks. I'm just presenting a couple of instances. Now to finish up, I just wanted to address a couple of caveats with the method and then sum up. The caveats which many of you might be thinking are ChIP-seq has issues as ground truth, there's cross-linking bias, there's antibody design. We need orthogonal ground truth and validation methods, but our conclusions are fairly broad, and I'm happy to talk about that. More held out cell types would be nice, different processing and features. And fundamentally, it comes down to this is a challenge which measures model performance. We'd like to understand the biology better, particularly when measuring in vivo cross-cell type binding. And so we'd like to incorporate data which more functional binding and convert this into useful biological insights, which was the purpose of this exercise. So to sum up, top performers do improve over the state of the art quite significantly, but as you saw from the raw numbers, there's a lot of work to be done to bridge the gap to feeding it to an experimentalist for downstream genome-wide validation in an unseen cell type. Performance measures and cross-cell type performance really do matter, as does background set, and just looking at what might be coming with in vivo binding, it's clear that fitting sequence sort of too well can hurt adaptation to a given cell type. We need methods that explicitly adapt to a known test cell type. There's no sort of universal predictor possible without knowing the test cell type. You need to know it, and then you can bridge that gap. And you need to account for these cell type specific sites, which often predominate. And I'd like to wrap up by thanking a lot of people who were involved in conducting this challenge, from ENCODE, DREAM, and Sage Bionetworks, who graciously hosted it, and to all the participants. Thanks very much.
Thanks, Akshay. One question, everyone? And Sean? You have a question?
What do you think to put this challenge, provide all the data set in Kaggle, so that all the data set community can try to attack this problem?
Right, so because of the tie-up with Dream, you can see the link there, the bottom right for the challenge hosting and data. It's currently there. That's Sage Bionetworks again. We are looking at moving it off there because, there's a rapidly growing platform of my analysis code for all this and a lot more, as well as visualization and all that for these. So we'll be looking at where to move it next. There's several options. We should talk about it offline.
Thank you. Let's thank Akshay once again.